Hello everyone, my name is Gary O'Mealy, and in this video we are going to talk about G-protein coupled receptors. So in the previous few videos we have talked about how pretty much every type of cell signaling is going to involve a signaling molecule, whether it's a hormone, neurotransmitter, or miscellaneous, traveling to a target cell which has a receptor for that particular ligand. So the target cell's eventual response, which we laid out in a couple of the previous videos, is going to be dependent on the actual mechanism of transducing and amplifying the signal. Now, in a previous video, we laid out basically three different types of signal transduction pathways. We looked at the direct opening of an ion channel, and then we also mentioned G-protein coupled receptors, which are going to be covered in this video, and tyrosine kinase receptors, which will be covered in the next video. So for now, our focus is going to be on the G-protein coupled receptors. G-protein coupled receptors are usually pretty easy to identify because they are 7-pass integral membrane proteins, and to clarify what exactly we mean by that, this is a membrane protein that has 7 transmembrane domains, meaning that essentially there are 7 portions of the protein that pass through the membrane and then loop back around to pass through the membrane again. So you can count these up: 1, 2, 3, 4, 5, 6, and 7. So, as a receptor, it has an extracellular facing ligand binding domain, and then the C-terminus is going to interact with the G-protein that we will talk about here in just a second. So, the G-protein, like I said, interacts with the intracellular facing C-terminus of the protein. So, you'll notice that there are kind of 3 different subunits to the G-protein here, so let's focus on that for now. So these 3 subunits are called G-alpha, G-beta, and G-gamma. Of particular importance to us for now is the alpha subunit. So the alpha subunit is a peripheral membrane protein, and most importantly, it is a type of enzyme called a GTPase. If you've heard of ATPases before, this is very similar, except this is a GTPase. It is going to bind to and hydrolyze a slightly different kind of nucleotide compared to ATP. So the deal with this alpha subunit is that when it is inactive, it binds to GDP, which is the low energy form of the GTP nucleotide. The beta and gamma subunits are regulatory proteins that bind to the alpha subunit when it is in an inactive state. So, the important thing that you'll notice here is that the alpha subunit is inactive when the receptor is not bound to its particular ligand. So, like I said, in the absence of the ligand, this whole setup here is going to be in a state of dormancy. It's going to be inactive, but let's see what happens when the actual ligand binds. So the ligand comes from the extracellular fluid, binds to the portion of the receptor in which it binds, and this is going to cause the G-alpha subunit to exchange the GDP for the high-energy GTP. This is going to activate the alpha subunit. It's going to undergo a conformation change, and this is going to cause the alpha subunit to separate from the beta and gamma subunits, and all 3 of them are actually going to separate from the G-protein coupled receptor itself. So this is the basics of activating a G-protein coupled receptor. If it seems very simple, well, it is very simple, but what's not going to be quite as simple is what happens after this. And we are going to see that there are actually several different types of responses that the cell can have depending on what the ligand is and what particular kind of G proteins that we're working with. But the important thing to recognize for now is that the alpha subunit and the beta gamma subunits are now going to act separately from each other. So I mentioned this on the previous slide, but there are actually many different subtypes of the G protein subunits. So you can kind of look into this on your own. It's not vitally important that you know that right now for this particular class. But basically, the combination of G protein subunits that a particular receptor uses is going to help determine what the eventual outcome of the receptor activation is going to be. So there's several different things we can do. We can open a metabotropic ion channel. This is a little different than the ionotropic one that we've seen before. We're going to, or we can rapidly generate second messengers, which are going to amplify your signal as we've laid out before, and it's going to help us produce much more complex, much more intricate intracellular responses. So let's take a look at what both of these look like. So G protein coupled receptors can directly open ion channels. So like I said, this is different than opening an ionotropic channel as we saw before. With an ionotropic channel, the ion channel is the receptor itself. So for a metabotropic channel, which is what we're looking at here, the channel is distinct from the receptor. You can see your G protein coupled receptor is over here. The ion channel we are eventually going to open is over here. So they are two different things in this case. So as you can imagine, in the absence of a ligand, the G proteins are inactive and the ion channel is going to be closed. In this particular example, we are choosing to make this ion channel a sodium channel. You will hopefully remember that sodium is maintained at a high concentration in the extracellular fluid compared to the intracellular fluid. So there is a strong concentration gradient that wants to pull sodium inside of this cell, but because this ion channel is currently closed, we cannot act on that gradient. So if we do bind a ligand to this G protein coupled receptor here, we are going to activate our G proteins, and in this particular scenario, the activated alpha subunit diffuses across the membrane to interact with that metabotropic channel, and that interaction causes the channel to open up, and now sodium can flow into the cell from high concentration in the extracellular fluid to low concentration in the intracellular fluid. The downstream physiological consequences of opening an ion channel like this is going to be explored in much more detail when we eventually start talking about the nervous system, so you can kind of look forward to that. Okay, so in addition to opening metabotropic channels like that, another very important thing that G protein coupled receptors often do is they activate second messenger systems. So there are several different kinds of second messenger systems that can potentially be activated by a activated G protein coupled receptor. So we'll look at two of the most common ones, but both of these are going to involve the active alpha subunit interacting with and binding to a membrane-bound amplifier enzyme. Okay, so this is an enzyme that is inserted in the cell membrane that is, once activated by the alpha subunit, it is going to rapidly start catalyzing a reaction that will produce a second messenger product. So the most common amplifier enzyme that we are going to see is called adenylyl cyclase, or adenylyl cyclase. I find adenylyl cyclase a little bit easier to say, but adenylyl cyclase uses as its substrate ATP molecules, and once activated by the alpha subunit here, following activation of the receptor, adenylyl cyclase is going to catalyze the conversion of ATP into a second messenger called cyclic AMP. Cyclic AMP is possibly the most common and most potent second messenger that you can find inside of a cell. So this reaction that you see here is very fast, and following activation of adenylyl cyclase, the concentration of cyclic AMP is going to increase from 0 to very, 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 very high levels in the intracellular fluid very, very quickly. So this is the amplification that we talked about in a previous video. Even if you start with an initially very low concentration of initial ligand, you can greatly amplify the signal if the message is conveyed intracellularly by a second messenger like cyclic AMP. Okay, so what is cyclic AMP going to do? Cyclic AMP can actually do quite a lot of different things, but one of the most common things it's going to do is interact with another enzyme inside the cell called protein kinase A. So in a previous video, we talked about the roles that kinases play in phosphorylating other proteins and changing their function to produce physiological effects inside the cell. So hopefully you will immediately recognize the significance of cyclic AMP interacting with a previously inactive protein kinase A. Now, the way I've illustrated this here is a little bit oversimplified. In reality, what happens here is that in the absence of cyclic AMP and in the absence, obviously, of receptor activation, protein kinase A is kept in an inactive state because it is constantly bound to a regulatory protein that inhibits its function. So what cyclic AMP is really doing is cyclic AMP binds to binding sites on that regulatory protein, pulls the regulatory protein off of protein kinase A, and that allows protein kinase A to essentially be unshackled so that it can unleash its phosphorylation activity. Basically, you can kind of think of it like cyclic AMP takes a ball and chain off of protein kinase A and, like I said, unshackles it. So once protein kinase A becomes activated, it begins phosphorylating a lot of different proteins and enzymes inside the cell, and the activation of these previously inactive proteins is going to be what produces the nature of the physiological response inside of this target cell. So we will see plenty of examples of these types of second messenger systems and what they specifically do in target cells in the future. So this adenylyl cyclase / cyclic AMP system, like I said, it's probably the most common way that G protein coupled receptors produce intracellular responses in the human body. So there is one other slightly less common system, but is still fairly common enough, at least that it's going to be worth our time to mention it. So let's go ahead and take a look at that. So this system that we're going to look at here starts very much the same way in that you activate the G protein coupled receptor, that activates the alpha subunit, it binds to GTP, it diffuses over, and it activates an amplifier enzyme. Well, what's going to be different here is that in this particular case, we are activating a different amplifier enzyme. This amplifier enzyme is called phospholipase C. It catalyzes a totally different reaction than adenylyl cyclase did. It's going to have different substrates and it's going to have different products that are going to act as second messengers in a different way than cyclic AMP did. So the substrate for this phospholipase C enzyme is phospholipids within the membrane. One of the most common phospholipid substrates is called PIP2, which stands for phosphatidylinositol bisphosphate. It's fine with me if you just refer to it as PIP2. So what phospholipase C is going to do is hydrolyze the PIP2 into two different components. Diacylglycerol, which is insoluble in water and is therefore going to stay associated with the plasma membrane, which has the hydrophobic inner core, and then inositol trisphosphate, or IP3, which is soluble and will enter into the cytoplasm and act downstream from here. So IP3 and DAG are both going to separately act as second messengers following their release from the PIP2 by phospholipase C. So first, let's take a look at what diacylglycerol is going to do. As I said, because of its insolubility, DAG will stay associated with the cell membrane, and it will diffuse over and activate protein kinase C, which is a membrane-bound kinase, which is going to produce somewhat of a similar effect that we saw to protein kinase A. We activate this kinase, it starts phosphorylating a bunch of proteins inside the cell, and boom, you get some kind of response. Now, what IP3 is going to do is totally different. IP3 will diffuse through the cytoplasm, and it will bind to a receptor that is on the membrane of the endoplasmic reticulum. Something we may have or may have not mentioned in detail before is that one of the major functions of the endoplasmic reticulum, in addition to protein and lipid synthesis, is that it acts as a storehouse or a reservoir for large amounts of calcium. Calcium can sometimes cause problems inside of cells because any phosphate compounds, including DNA, will become very insoluble in water if they interact with calcium. So for that reason, the cell intentionally keeps calcium levels in the cytoplasm near zero. And the way the cell accomplishes this is by making sure that pretty much all the calcium inside the cell is sequestered inside of the ER. However, calcium can be very useful in signal transduction processes. So what IP3 is going to do here is bind to this receptor, and that is going to trigger the opening of a calcium channel in the ER membrane. This will cause calcium to diffuse from its area of high concentration inside the endoplasmic reticulum to low concentration in the cytoplasm. And from here, these calcium ions can act as very potent second messengers themselves. And usually what they're going to do is interact with calcium-binding proteins like calmodulin, which are then going to produce downstream effects as well. We're not going to go into any further detail on that for right now, but we will see some examples of that in the future. And if you want something to look forward to, you can go ahead and look forward to the fact that calcium release from the endoplasmic reticulum is going to play a major role in getting muscle contraction to happen. So we will definitely talk about that in the future as well. Okay, that's going to do it for this video. Here is a list of vocabulary terms you probably ought to be aware of. And for checking your understanding, number 1, how does the enzyme activity of a G protein determine whether it can activate downstream targets like pKa or PLC? Number 2, what roles do kinase enzymes play in the function of G protein coupled receptors? And number 3, what kind of previously discussed process could lead to all of the cell's calcium being stored inside the ER lumen? Alright, that's going to do it for this video. If you have any questions, go ahead and drop them in the comments section. Otherwise, I will see you in the next video. So long.